Okay, so ladies and gentlemen, you are all welcome to my YouTube channel. Today we are going to look at the concept of genetic disorders. And under this, we are going to look at what are the genetic disorders, and then why do they occur, and then who is affected with the genetic disorders. So we are starting with the concept of mutation. So what is mutation? Mutation is, of course, we need to know that mutation is usually occur either in a chromosome or in a DNA. So, of course, remember from our basic knowledge of biochemistry and biology, we know that nucleotides, nucleo, nucleotides, when nucleotides joined together as a building block, they generate what you call a DNA. So when we have nucleotides as a building block, the nucleotides as a building blocks joined together to produce what you call a DNA. Like, for example, in a DNA, you can have millions and billions of these DNA of these nucleotides. Like, for example, in human being, we have like about 3 billions nucleotides that construct to form the DNA. So what is a mutation? So a mutation is a change in a nucleotide sequence, the change in a nucleotide sequence of the DNA. That is mutation. Assuming now you have a sequence of nucleotide, let's say you have A, C, G, T, G, C. So assuming now this is the nucleotide sequence. So if this nucleotide sequence is changed, like for example here, in this nucleotide sequence, let's say here we have T. So if this T is changed to, let's say, maybe when it's changed to, let's say, G here, it means that there's a change in a nucleotide in this sequence. So the change in a nucleotide sequence is what you call mutations. And this mutation can be positive or it can be negative, so that it can actually be positive or negative. So now let's look at the causes of mutation. So what caused the change in these nucleotides? So the causes of mutation include exposure to toxic substances. So when you are exposed to toxic substances, it can actually lead to the change in a nucleotide. And of course, this exposure to the toxic substances include radiation and then chemical substances. So ultraviolet rays and X-rays are the examples of radiation that if you are too much exposed to them, they can cause mutation in your DNA. And of course, we have chemicals such as PCBs and DDT, which is actually insecticides. So these chemical substances, PCBs and DDTs, they can also cause a mutation. And there are so many examples of the chemical substances that cause mutation. So like for example, any substances or any material that have the potential to cause mutation in a living organisms, that substances, or to cause mutation of a DNA in any organisms, that chemical substances are called mutagens. They are called mutagens. So any substance that can or that have the potential to cause mutations in any DNA sequences of any organisms is called, or they are called mutagens. So here the PCBs, DDTs are examples of mutagens. So the mistakes or these mutations is usually, although the mutation can be occur naturally, it can be occur naturally. So this mutation can occur naturally. So like for example, during meiosis, this mutation can also take place. And the mutation can happen at random. It can happen at any position. It can happen at any stage. So, but usually these mistakes of mutation is usually taking place during meiosis. So we should understand that during meiosis is usually where we have the mutation. And in addition, these types of mutation, remember that from our basic knowledge of cells, we have two types of cell. We have two types of cell. We have that is based on the types of the cells that are found in humans in terms of the specialization. So we have what we call somatic cells. We have what we call somatic cell, somatic cell, and we have what we call gamete cells. We have gamete cell. So somatic cells are the body cells, while gamete cells are reproductive cell. So if mutation is actually occur in somatic cell, so that types of mutation cannot pass to the young ones. It means that it cannot be inherited. But if mutations occur in the gamete cell, so these types of mutation can be actually passed on to the young ones, so it can be inherited. So that is somatic mutation and gamete mutation. And then the next thing that we are going to look at is what are the outcome of mutation? If these mutations occur, what are the outcomes? So the outcomes, the mutation can be negative. It can have a negative effect. Like, for example, change in the DNA sequence can lead to wrong protein produced. Like, for example, from our basic knowledge, we know that the DNA produce RNA and then the RNA produce protein. So if there is a mistake in the DNA, it means that the RNA will have a mistake. And of course, if there is a mistake in the RNA, of course, it will occur in the protein that will be produced by that RNA. So whenever there is that problem of protein that is produced finally from the mutated DNA, there is a likelihood that a disease or disorder will occur. And this disorder is uh, is called genetic disorder. It is called genetic disorder. And there is also a mutation that doesn't have effect. It doesn't have effect. Like, for example, in the human DNA is made up of different components. Like, for example, in part of the DNA that we have what you call a junk DNA. And when we say junk DNA, it means that it's a part of the DNA that even their function is not known. Their function is not known, and most of this part of the junk DNA is not actually involved in producing protein. So no proteins is produced. Even the functions of that junk DNA is not well known, although it has been established in some research that the junk DNA have a regulatory function. But generally, if the mutation is occurring in or it occurred in the junk DNA, it doesn't have any effect because the junk DNA it does not code for anything, so it doesn't code for any protein. And then the next thing is another effect of or another outcomes of the mutation. It can cause positive effects. Like, for example, there are some cases that it can promote some characteristics that allow beta survival, and that is mutation. Sorry, evolution. Because remember, from the concept of evolution is when you have an organisms, maybe the environment is changing, and of course, those organisms that cannot survive the environment, of course, they will die. So those organisms that can survive, it means that they will be able to actually promote some characteristics or some features that will allow them to survive in the environment. And that is the survival of the fittest. So that changing that actually promotes some of the organisms to survive as a result of the change in the environment. So it's usually come through mutation. So mutation is what leads evolution. So it is a positive effect because it allows the survival of the living organisms in the environment. So we have types of mutation. we have types of mutation, and these types of mutation, we have what you call substitution. We have also deletion, addition, and inversion. Like, for example, these are the sequences of the nucleotides starting with A, B, C, A, T, C, T. So when we said substitution, it means that substituting a base. So you remove one base and substitute it with another. Like, for example, here, this is adenine, thymine, cytosine, adenine, guanine, cytosine, and thymine. So if, for example, here is C, here we have C, but you see that at this point, the C here is at position 3, but we can clearly see here from here, the C that we have here is now substituted with G. So this is what we call substitution. So when one nucleotide change or substituted with another nucleotide, or when a base is substituted with another base, that types of mutation is called substitution mutation. Like, for example, these types of mutation is what even actually leads to even a disease called sickle cell anemia. It's due to substitution in the hemoglobin gene. So sickle cell anemia is due to sickle cell anemia. Sickle cell anemia is due to the substitution mutation. And then the next types of mutation is deletion. So when a particular nucleotide is deleted, so that types of mutation is called, or when a base is deleted, so that types of mutation is called deletion. Like, for example, look at it here. We have ATCA, TCT. So we have T here. So the T here is being deleted. So that is why T is missing. Like, for example, here we have 1, 2, 3, 4, 5, 6, 7. Here we have 1, 2, 3, 4. We have, okay, 1, 2, 3, 4, 5, 6, 7. But here we have 1, 2, 3, 4, 5. So it means that the T here is deleted. This T here is also deleted. So these types of mutation is called deletion. And then there is an addition. Like, for example, here we have ATC, but here we have another C and we have another G here. So these types of mutation is addition, because some nucleotide or some bases are added which are not present in the original. And then we have inversion. It means a flipping around a sequence of 3 bases. So if there is a flipping, like, for example, here we have ATCA, GCT. So now we can clearly see that this is where we have the C, but it's changed here. And then we have A. The A also changed to here. And then, of course, we have G, which is still remain here. So these types of mutation is called inversion. Some nucleotides are flipped to another position. And these types of mutation is called inversion. Then the next thing is the genetic disorders. What are the genetic disorders that are caused by this mutation? So how do they occur? Sometimes a chromosome fails to separate correctly during meiosis. So this is called a non-disjunction. So, for example, during meiosis, the nucleotide tend to separate. Like, for example, look at it here. We have meiosis 1 start normally, which is we have tetrad line of in middle of a cell. So when you have a situation where all the gametes have abnormal number of chromosomes, either 1 or too many or 1 few. So this is actually happened when the chromosomes are failed to separate correctly. So this types of condition is called non-disjunction. Like, for example, a sickle cell anemia. A sickle cell anemia, there is a red blood cells that have a shaft like C, so it doesn't carry enough oxygen. And of course, the red blood cells clump together, and it occurs in the African and American population. And one of the signs and the symptoms of the disease, there is a tiredness, is often a symptom. So as I said, this sickle cell anemia is also another types of diseases that affect humans and is actually due to mutation. And another disease is Down syndrome. So Down syndrome is when you have an extra chromosomes on 21. So there are supposed to be two chromosomes. So when you have an extra chromosome there, then it now changes to from 2 to 3. And of course, the condition is called Down syndrome. And of course, the people that have this problem with the extra 21 chromosome, they suffer mental retardation, the short stature, and then cement crepes on farm like this here that we can see clearly from the picture here. That is the cement crease on farm, and then there is an efficient fold in eye, as you can clearly see here. So these are the symptoms on, of course, the problem associated with Down syndrome, and it's one of the genetic disorder and is due to mutation. And then there is no cure for Down syndrome, but you can actually try or you can test the disease through what you call prenatal tests, such as cryotyping, triple screen, blood test, and many more. So these are the ways that you can test for the disease. So it includes cryotyping, triple screen, and blood test, which means that you can test whether a particular child is having Down syndrome or not. And this is going through typing and triple screening as well as the blood testing. And then in addition, we have amniocentesis, which actually allow the parent to find out before child is born and prefer emotionally. So Down syndrome patients have symptoms such as heart abnormalities, small skulls, extra fold of skin under the eyes, large protruding tongue, and a flattened nose bridge. So these are actually the conditions associated or the genetic disorder. Like for example, this amniocentesis is actually a way of connecting or collecting amniotic fluid from a baby and then test it whether the baby is actually having or is developed with the Down syndromes or not. That is the Down syndrome. So when you have an extra 21 chromosome, there's also the Klinefelter syndrome. So Klinefelter syndrome is when there is an extra X chromosome. So when there is an extra X chromosome, like for example, generally male have XY chromosome. So in female, when there is an XY, when there is an extra chromosome, they are going to be XXY. So this is a male who has underdeveloped sex characteristics. They have this problem and still they cannot reproduce. They are infertile. So this is actually some of the condition. So that is for the Klinefelter's syndrome. They have tall stature, narrow shoulders, and they also develop gynecom, okay, gynecomastia, and then there is also small testes and infertility. So these are actually the signs and symptoms associated with the Klinefelter's syndrome. So they have 47 chromosomes with extra X chromosome. So then the next thing after that is actually what you call human cryotype. So you see, this is a cryotyping. So when there is an X chromosome, extra X chromosomes, as you can clearly see here, that is when we have the Klinefelter syndrome, and this can be actually done through cryotyping. That will give you an idea of the exact number of chromosomes. And then, so the next one that we are going to look at is Turner syndrome. when there is only one X chromosome. So this is a female with underdeveloped sex characteristics. So they are characteristics of this. So they only have one X chromosome in the case of Turner syndrome. And this is a female with underdeveloped sex characteristics. And of course, the characteristics include that is the symptoms include short stature, webbed neck, broad chest, increased weight gain, and then of course steroid. So this is actually the case of the uh Turner syndrome. That is, they have only X chromosome. You see, so this is how it is using cryotyping. Only have one X chromosome. So other genetic disorders include we have achondroplastia, that is dwarf, cystic fibrosis, hemophilia, progeria, and Tay-Sachs. So all these are the examples of the genetic disorders. Ladies and gentlemen, this is where we are going to stop. And of course, if there is any video, then you can ask. Sorry, if there is any questions, you can ask on the comment section. Thank you for listening. And of course, if this is the first time you are coming, please subscribe to my YouTube.